Okay, so today is going to be the last pedagogical lecture, and there are going to be two parts. I'm going to cover the material in Chapter 12, which is sort of a broad overview of development and aging, sort of the full lifespan. And I'm shooting to talk for about an hour and a half or so, and then we'll take a little break, and then Jesse will come in and give some more specifics about aging. Okay? So again, we'll cover behavioral processes, brain substrates, and clinical perspectives, and start by talking about the developing memory in infancy through childhood. So some of the reasons why we're interested in the development of learning and memory: ideally it should help us tailor education, help us understand normal brain development. These are some of sort of the broader goals. But we also need to be aware that these developmental trends represent just averages, and there's a huge amount of individual differences. It's hard to talk about what's normal, and that a huge amount of variability across and within people. So learning starts even before birth. I'll have to ask Jesse to give some sort of personal insights on this, but you can train a fetus to recognize sounds. So by 3 to 4 weeks, 34 to 36 weeks, so that puts us at what month there? Divide by 4. The end. That's towards the end. That's towards the end. So you put a speaker on the mother's abdomen. We could have actually done an in-class demonstration here. It didn't even occur to me.
Not quite there.
Well, but we could see if we could move this earlier. Sounds initially cause fetal movement, and repeated sounds the decreases the response. So we basically see the habituation that we talked about, and you actually see it in the fetus. So you can also see prenatal preferences formed that persist after birth. So the first one is just completely done in utero, looking at habituation measured by kicking. So mothers read specific books twice a day for the last 6 weeks of gestation, just what you want to be doing. 2 days after birth, babies are given an opportunity to work to hear the familiar story or a novel story. And a long pause in sucking gives them familiar. So basically, this is a way of, obviously, 2 days afterwards, we're basically using the same procedures you use in animal conditioning, because obviously these are nonverbal. And so the idea is that if there's a long pause in sucking, they get the familiar story. If there's a short pause in sucking, they get a novel story. And for half the babies, you reverse it. Babies like familiar stories, as you'll find out from reading the same bedtime stories over and over again. And regardless of contingency, babies adapted sucking rates to gain exposure to the familiar story. So even just reading them this story for 6 weeks, by the 2 days after they're born, they recognize the story that they were read to. So moving past sort of in utero training, infants demonstrate a variety of learning that comes from language, motor skills, social skills. But some of the questions are sort of what are the trajectories? To what extent are the trajectories? What modulates these trajectories in which they learn? Some of them have to do to immature sensory and motor systems. Some have to do to immature brain circuits. But one thing that's shown is you can see classical conditioning. Let's see if this should jump automatically. Oh, this was working before. Okay, hold on 1 second. Maybe that was working. This is a long
What I'm going to be doing is tying the ribbon around Jessica's ankle, and then I'm going to be having her go with her crib onto the stand that doesn't have the ribbon attached. Ready to play? And the way we try to find a kick is when the leg goes out and then comes back in. Write down all the kicks for every minute. And this is my first one. And now I'm going to attach the ribbon to the mobile. And as Jessica kicks, it's going to move the mobile. onto the empty stand. And this is a little test we have just to see if the baby has learned.
So, oops. So this is work actually done here in New Brunswick, at Rutgers in New Brunswick. So basically what you're seeing here is instrumental conditioning, where kicking the leg is moving the mobile. The mobile is positively reinforcing and leads to vigorous leg kicking. And the memory can be maintained for days with no reminders. And they do all sorts of training paradigms like we've seen before in the instrumental conditioning section, where you have different crib liners and so forth, so that you sort of learn contextual. So you can do all sorts of conditioning studies as well. You can also do classical conditioning, okay? Remember the baby Albert studies from before. And the basic aspects are intact. You can get eye blink conditioning, like we talked about, but it's often slower in infants. But trace conditioning, which is where there's a gap between the end of the CS and the beginning of the US, which is primarily used depending on hippocampal function, not possible until age 4. So you can use these different paradigms to begin to look at some of the ways in which different brain systems are active. Another form of learning that goes on both in the laboratory and the life is social learning. And you can find that infants, even at 2 to 3 weeks, exhibit the imitation of facial expressions. Okay, so this may be, and the question is, you often see, I should have got an image here of, people who are making little faces at babies, and the question is, who's conditioning who? If you remember, we had some issues before about kids in the bed, and it wasn't clear that the kids were conditioning the parents, and the parents were conditioning the kids. So when people are making funny faces and the babies are making those faces back at them, I think they're sort of mutually conditioning each other. So one of the big questions is about whether or not babies have episodic memory. And this is the same question that came up when we talked about some of the animal data on facts and events. It's hard to assess episodic memory in both an animal and an infant because you can't ask them, do you remember what you had for breakfast or what happened yesterday? The question about semantic memory. Clearly children are learning facts and events. Or learning facts and relationships. So one of the ways in which this is studied in infants is you have an elicited imitation paradigm. So children are shown how to operate a toy puppet, okay, or how to operate a puppet alone, and then they're presented with the same puppet, and children who have seen the puppet demonstrated were later more likely to see it and to use it. So some of this sounds somewhat trivial, but the idea is that children at various ages, you can sort of see whether or not they can learn from observing somebody else doing something. This is the sort of imitation. Do they remember episodes? Again, if you remember the scrub jay experiment, where there's a whole question about whether or not these birds can remember specific events. And the way that was done was noting not only do they know where food is buried, but they know what was particularly buried where because they know what will have gone bad. So here, one of the things that the children are learning various question-answer pairs, okay, and later tested for where they remembered where they learned it, what was the source of a fact. So there's distinguishing between a paired associate and the specific context in which it was learned. And what they found is that young children have a very poor source memory. So the idea, this is where they may be sort of being to learn information, but a poor memory for sort of when and where it was actually learned. Okay. One of the big questions, a lot of the areas where small children are impaired or less relative to sort of older children have to do with hippocampal function or tasks that we know depend on hippocampal function. So one of the questions is, are some of these due to slow hippocampal development that's needed to support episodic memory? Is there something about a developing sense of self? Do people know what the red mark test is? Does anybody know what that is? Somebody want to explain?
It's when they would mark the child with something like something that was, like they would look in the mirror to see if the child touched that mark, so they know that that's the ultimate.
Right, right. You see that it's often done in animals as well, apes and so forth. They show they have dolphins can do it too. Dolphins know it themselves. So, or is it just that they have an episodic memory, but they can't express it because we generally express our episodic knowledge verbally So a lot of uncertainty in towards why it's so hard to see evidence of episodic memory in small children. Big aspect of development is the question of sensitive periods. So some learning abilities are only available early in life. That's called a sensitive period or a critical period. Those are generally synonymous, after which some forms of learning become difficult or impossible. Let me see, does this video start here? I don't know why this is not working. Okay. So there's just a little brief overview of Conrad Lawrence and critical periods in imprinting. So the basic idea, this study is just one of these classic studies. The images of the ducklings and Lawrence are sort of classic in varieties of psychology. And the critical idea being that you can imagine from an evolutionary point of view that it makes sense to develop this sort of imprinting sort of on the first person that you see when you're born. In this case, in the ducklings, it's really within the first several hours that it has to be. So it suggests that there was this, not only does it suggest there's sort of an innate predisposition to sort of learn to imprint on someone who takes a sort of a mother role, but also that there's a critical period when it can happen. So imprinting is forming a close bond with the first individual seen at birth. It's common in birds, but also occurs in other species. Bonds are best formed immediately after birth. After this critical period, it's harder to form. We see sensitive periods or critical periods in a variety of other systems. In visual development, if you have an impaired visual input early on, it leads to permanent disruption of the visual system. There have been a number of classic studies with cats, for example, that are reared in vertical only or horizontal only visual environments, and that this sort of disrupts their visual system permanently if they don't see a wide variety of vertical and horizontal lines. If birds are raised in isolation and not exposed to other songs, they lose the ability to sing. They can't be learned later. So a lot of suggestions that for different types of learning, there are very critical periods where you need to be exposed. And it sort of sets up an overall sense of ourselves being wired, in part based on, sort of evolved to be hardwired to expect certain things to happen developmentally, experientially. And that the combination of what we're sort of wired to learn and what we're exposed to need to occur in combination. Obviously, much of this has been sort of suggestive of language, that language also may have a critical period. Some people suggest that if you don't learn any language before 12, you'll never master it. There have been occasional cases, what are called feral children, children who are sort of raised without language, and they prove unable to learn language. But of course, these are obviously not controlled studies. Nobody that I know of is going to volunteer a child to be a random controlled study of lack of language exposure. And these few children who have been raised without language, you don't really know why they were, they may have been left alone in an attic for 12 years because they were retarded and nobody could deal with them. So, and this just shows infants distinguishing. But one of the ways in which we do see something like a sort of phonemic specific effects have to do with the development of sound distinctions. Okay, if anyone's ever seen somebody, somebody Japanese ordered fried lice, and no matter how many times they say, no, not fried lice, it's fried rice, they still say, yes, that's what I said, I'd like a bowl of fried lice. And the reason is that a lot of these phonemes, these distinctions, disappear after a certain point if they're not reinforced. And this shows, I don't even know how to say these. Ta. What's it? I don't know how to read this kind of ta or ta. I don't know what the difference is. Do you? So, yeah. Uh-huh.
The sound, the difference between V and W doesn't exist there.
Uh-huh.
So we'll have, when I used to teach.
It's very nice to see you.
Yeah, there's
Right.
no V. Everything is W.
That's why I said it's very nice to see you.
Yeah, kind of. And so like the little kids will come in and they can't say van. Like
Uh-huh.
I used to teach little kids how to read, and
Uh-huh.
like even going the van, they can't do it because
Right.
it's just not a sound that exists.
So that they use the W. So that's another example. Adults can learn a second language more quickly than young children, or they don't usually end up at the same level of mastery. It's pretty disheartening to go and see these like little 3-year-old French kids having absolutely perfect French accents, and you could study for years and you don't get it. So some of this is obviously due to time on task, but obviously there's also a sense of an exposure that kids, you see it when I had a postdoc who came to visit from Israel, and her kids spoke no English, and they were all about this high or so, and she came into the lab within like the first week or two practically in tears herself, because all 3 of her kids were in tears because they don't understand anything, they're miserable, they're unhappy, they can't understand anything in the class. And I says, mark my words, I says, in 6 months, the kid's biggest problem is that they're embarrassed that mommy doesn't know how to pronounce words. And sure enough, 6 months later, the kids were sort of correcting her pronunciation that she could never get right on various things that the kids did. So they absorbed quickly. Let's talk about the adolescents. I had the pleasure and challenge of taking care of my 13-year-old niece and her best friend for the weekend. And I can certainly verify that it's a challenging period. So transitional stage between the onset of puberty and full adulthood. This is particularly an area where working memory changes are taking place. Executive function, digit span increases through the early teens. Executive function develops throughout early adulthood. So this looks at digit span, so how many digits you can keep in mind in memory. We can see it sort of continuing to rise up through 15. Here, the 2-back task, one of these classic N-back tasks that we talked of as a marker for frontal function. And we can see with a high degree of variability, it's continuing to increase up until the early 20s. Thank you. So it clears this ongoing development of the frontal lobes goes up. What is that? Ongoing development of frontal lobes. This is probably also linked to why some frontal lobe disorders don't show up until college, like schizophrenia. Typical first episodes of schizophrenia are often in high school or college. Let me just move there. So let me talk about another issue. Let's take I'm going to show you a bunch of, move to another topic. I'm just going to do a quick thing. So let me just have everybody shout out as quickly as possible. I'm going to show you some stimuli, and I'd like you just to shout out as quickly as possible, same or different. Okay? Shout.
Same.
Okay? So these are objects that are rotated in 3 dimensions. Okay? I didn't hear everybody shout out at the same time. That's what I want to hear. Okay?
Same. Different. No, different. No, those are different.
No, that one is rotated this way, and
They're
this way.
different.
Okay. So we have actually a pretty good gender balance in this class. So these tasks developed by Roger Shepard, who was one of the people I studied with at Stanford, called the Shepard and Metzler task, and it's the task for which you probably see the biggest variance between men and women in the tasks that involves looking at something and rotating it in 3 dimensions in your head and mapping it to something else. Okay, and what?
Dr. Tracy says that's why men are better at ortho class than women, because they can rotate the mouse.
Okay, could be. So broad ranges of gender differences. So there are many factors that affect the sort of the lifespan trajectory. Women perform slightly better on average on verbal memory and spatial learning of landmark locations. Men perform slightly better on average on spatial learning routes, and they keep putting this on average just because someone's going to say, but wait, I can do this. So a lot of strong emotional feelings about a lot of these gender issues, as Larry Summers' impolitic remarks a number of years ago brought to the fore. Similar gender differences are evident in many mammals, suggesting the differences aren't purely cultural. So, for example, you find that in rats, you see male rats tend to navigate more by dead reckoning, and female rats by landmarks. These sort of verbal type differences, you can't measure those in animals, of course. So the spatial tasks are easier to measure in animals. Here, where there's obviously always a question with gender, is it sort of culture or biology? Some fascinating studies have been done looking at transsexuals before and after hormone changes they take, and you find that when you take men to women, that their verbal abilities improve, and women to men that they get worse. So it suggests a lot of the gender differences are probably due to both in utero differences in brain formation, which are also controlled by hormones, but also hormonal differences just in terms of fluctuating hormones, enough so that even some of these verbal differences may be as much attributable to sort of hormonal differences. I'm going to skip over this, but this is just sort of a summary of some of the different types of learning. We know about these types of learning in their critical brain regions. That's what we've been sort of studying all semester, and it suggests sort of when this sort of the timetable for development, that these areas are present, and when they're sort of developing later on. Let me just talk a bit now. I'm going to preview a little bit about aging. Jesse's going to talk more about it later. But this is a somewhat depressing graph for those of us who are somewhere in the middle there. This is why we're trying to get a lot of published done in the next 10 years or so, Mohammed, because then I'm going to hit this part here. So adult abilities can be stable for many years. Unfortunately, the overall pattern of decline in many things, Jesse will talk more about this a little bit later. Let me just skip over this for a second. The compensating thing is a lot of memory retention has to do with how you can link it up with what you already know. So people who are older may be a little slower in terms of sort of the raw processing, but have a richer network of past experiences so that when something comes in, you can link it to half a dozen different things you already know, sort of anchoring a new experience, while someone who's younger may only have one thing that they can link it to. So there is some compensation. So for example, older typists may make slower movements in terms of sort of raw processing speed, but have better memory and less need to look at manuscripts. So you can see, again, some compensating with sort of skill and knowledge and experience can compensate for some of these sort of raw. So overall, there's a lot of evidence that memory and learning a memory function is like a muscle in the sense that you have to use it or lose it, and that overall performance is preserved only in people who are actively engaging. And so the number one advice to people about keeping their memory sharp is to constantly be challenging it, whether that's playing Sudoku every day, or people always are asking me, what can I do to keep my memory sharp? And I say, learn a new language. I think learning something new, that not just doing the same thing over and over again, but learning new skills and doing it while jogging. So let's see. So the ability to retain and retrieve memories shows little decline. So people often say, Grandma, why do they say she has a memory problem? She just never stops talking about her childhood in Vilna and so on and so forth. And that's because these old memories are fine. What we see mostly in aging, a lot in aging, is hippocampal function, although Jesse will talk about some of the trajectories. What's often impaired, particularly late in aging, is forming new episodic and semantic memories. So to summarize, learning and memory ability varies over the lifespan. In infancy, we can see evidence of habituation, instrumental conditioning, classical conditioning. Semantic memory seems intact but developing at an early age. Strong development of working memory and executive function with some gender differences during adolescence. And after adulthood, a period of stability but overall decline in many basic and memory skills with different trajectories depending upon the brain systems involved. And some learning exhibits sensitive periods, such as imprinting. So let me turn now to talk about several factors in the brain bases of memory. Tremendous variation in learning and memory. How much of this is due to genetics? And I think Jesse will talk a little bit about that, right? At least some of this comes from studies of genetic mutations, selective breeding, primarily done in animals, and twin studies. So we'll look at each of these in turn. So here's one example, the BDNF, which I think Jesse may talk about more of. This shows that looking at the different variations, delayed recall of the BDNF, which is the brain-derived neurotrophic factor gene, that depending on which of the variants you have, that has an effect on your ability to do delayed recall, something we know is very dependent on the hippocampus. Oops. So here the val allele is the most common. The met allele, 1/3 have a 1 or more. And the more mets that you have, the worse the delayed recall. There's several other mutations. In animals, where one can sort of selectively breed, you can demonstrate genetic influences by taking rats on a maze, testing them for memory, and then those that do well, you breed them with each other. Those do poorly, you breed with each other. And that what you eventually get is a sort of a genetic separation. This is called assortative mating, or selective breeding is when we talk about rats. When it occurs in the natural environment, it's often called assortative mating. Can you think about how some of this may play out in the natural world? Obviously, we don't do these sorts of controlled studies. Where might you see evidence of this in the real world? Assortative mating, this kind of assortative mating and the effects. No, humans, I mean. Can you think examples of where this might happen? Where might this sort of issue come up in terms of humans?
In the workplace, like people they get, or that attend conferences together, that meet each other there. And
Right, so this has come up a lot. There was a very controversial book a number of years ago called The Bell Curve. Its main thesis was that in older days, we tend to sort of marry sort of within our group and so forth, and there were many things, you would marry, the president of the company might marry a secretary or someone and so forth. There was a lot of sort of much more sort of cutting across different, in this case really mostly about intelligence. But that as our society has become ever more sort of screening, particularly at the college level, for IQ, and much less about who your parents were, but more the sort of intelligence testing to get into schools, and the idea that college is, in a large sense, among other things, a place where many people will sort of meet and mate, that you get sort of more and more sort of assortative mating, that the people will tend to sort of marry people they meet in college. If they come from a very selective college, they meet other people, and so you begin to get sort of separations, that a lot of these sort of intelligence screening for admission to things like college and elsewhere, where people then are likely to sort of meet and mate, is leading to sort of more kind of distributions of intelligence. And that was the sort of controversial claim. This harks back to, do you remember someone from chapter 1 who sort of talked about this? This is about eugenics. This is sort of partly the idea. This sort of, there was eugenics, which was sort of eliminating the potential for breeding of those who are perceived to be sort of lesser on some scales. But positive eugenics, which is sort of negative eugenics, positive eugenics is something that sort of promotes the people who sort of have desirable attributes from sort of breeding. So this is sort of one way of seeing some of the effects of that kind of assortative mating on any sort of genetic linked trait, whether it's intelligence, which has some genetic component, or in this case, it's sort of maize bright and maize dull. Twin studies also demonstrate genetic influences. Generally what you do is you identify twins who are either fraternal or identical, test basic learning and memory skills, and see if genetic similarity is related to similarity in test results. Overall, genetic similarity predicts similarity of learning and memory skills. So identical twins tend to be the most similar, fraternal twins moderately similar, and non-twin siblings less similar. This has led to sort of broad estimates that about 50% of the variation in learning and memory may be related to genetics, although talking about the contribution of genetics as a fixed percentage is sort of not particularly meaningful unless it's a particularly narrow focus. I mean, genes and environment don't interact in an additive way, so you can say 50% this and 50% that. So it's often a much more complex interaction. But clearly, there's a lot of genetic component. Let's talk a little bit about the neurons and synapses in the developing brain and how this contributes to lifespan changes. The human brain is strongly influenced by the environment. So one of the features that we see, again, we talked earlier on about one of the reasons we talked about evolutionary theory early on, is that the ideas of evolutionary theory keep reoccurring in neuroscience and psychology in different ways. And one of the ways that evolutionary theory has influenced our understanding of the brain is seeing about evolutionary-like competition at the synaptic level. So we've talked about evolutionary competition, at the species level, and different birds with different beaks competing to sort of survive on a small island. We can see some similar analogous situations taking place at the neuronal level, where synapses and neurons will compete to survive, just the same way that a finch might compete to survive. So some of the ways in which overall that we see this happen is we see an overproduction of neurons. So often we see that there's more neurons than we need, more synapses than we need, and then there's a competition for the limited resources, and some will die off. So as I mentioned, this is very similar to natural selection, but what we're doing is sculpting brain development for maximal utility within an individual's environment. Okay. And we see this kind of competitive brain development, the survival of the fittest neurons, mostly in mammals, but we also see it in other animals as well. Some basic facts: neurons are produced very rapidly after conception, up to 250,000 per minute, which is just sort of an amazing number. By 25 weeks, production is mostly complete of sort of ramping up for neurons, and many, many more neurons are produced than needed. And so at that point, one begins to see sort of a competition. And we don't clearly know what this competition, the rules of it. Some resources, such as this brain-derived neurotrophic factor, seem to be critical. And there seems to be a general sense of sort of use it or lose it, that the more neurons and synapses are used, the more likely they are to survive. Okay? So the question is, if we map from the evolutionary theory to sort of neurogenesis, it's not clear what does it mean to be sort of survive. What is it that makes surviving? It seems to be that in some sense survival, to a first approximation of a neuronal element, a neuron or a synapse, has to do with its utility, how much it's used. Unsuccessful neurons undergo apoptosis, or programmed cell death. So up to a third of the neurons that are created may then sort of die off. And so this enables sort of cell growth to interact with environmental demands to sort of determine which is most useful. Synaptic connections started around 5 months, and rate about 40,000 per second are being created. More synapses are produced than needed, and the competition ensues. And what we see here is that many synapses are created, and unsuccessful synapses, those that don't play a critical role, tend to be broken up and material recycled. Okay, so life and death. And this competition and remodeling continues throughout life, although the pace and the scale can change. But the basic idea is that this overall competition between cells and within cells is producing a sort of an environment-driven sculpting of the neurons and the cells that are created. So again, we'll come back to that challenging period of adolescence. Adolescence corresponds to changes throughout the brain, but it's especially the frontal cortex, the executive. One sees strong synaptogenesis in the frontal cortex, so between 11 and 15. Okay? Myelination, the wrappings of the neurons that affects the efficiency of communication. Cortical axons are wrapped in myelin, improving the speed and fidelity. It starts after birth, but it's not complete until about 18 years, especially again in the frontal cortex. So we see over and over again that the frontal cortex, much of it's not actually functioning until you get to college. As well as big increases in dopamine input. So another aspect of frontal function that seems not to fully mature is the ways in which dopamine is modulating the frontal lobes. Okay? It's also adolescence is when the sex differences fully emerge, although they may begin before birth. Overall, the male brain weighs a little bit more, And some specific brain regions are larger in men. This shows us some of them, and others are larger in women. And so we see overall that it's not so much the case that men or women have any specific differences. And these brain differences may relate to some subtle differences in learning and memory that we talked about before. And one could have an entire lecture on sex and gender differences in brain function, so we're just sort of touching on it here as it relates to brain trajectory. The final stage, aging is often associated with brain deterioration. We see decreased neural density, synaptic density, overall loss of brain matter, up to 5% in weight by age 80. Aging associated with deterioration. Some of the loss may be due to neurodegenerative disease, namely Alzheimer's or mini strokes, but even in healthy aging, there's often loss of neurons and synapses. And Jesse will talk a bit more about this. We see in monkeys especially a decrease in the frontal lobe density as it relates to working memory performance across the late stage. Skip here. One of the ways in which we can understand changes in aging in neurons is looking at the flexibility in their encoding. This is rats are placed in a hippocampal maze. This is just a figure 8 shaped maze, and different neurons. Different neurons are colored, are assigned different colors, and so the fact that all this is sort of magenta is a particular neuron. It says there's a particular neuron that's firing whenever the rat's here. So this is sort of the notion of a place cell. The different neurons are selected to different places in the maze. And what you see is that when you bring the rats back at a later date, the same neurons are conserved. So the place cell for this sort of upper bar, that same neuron is also encoding the same place there. In contrast, for an aged rat, which develops place cells, if it comes back at a later session, it almost completely remaps. So the idea is that there isn't a lot of savings from one time to the other. So the older rats have to essentially relearn the entire maze. What's going on here? There used to be a belief that all the neurons you're ever going to have, you have at birth, and that these then sort of die off. We now know that's not true. There's evidence that suggests there's limited neurogenesis in adult mammals, and these are studied by injecting a radioactive tracer, which gets incorporated only into new cells, and then being able to image the cells that have been created since that tracer was introduced. And we now believe that there is neurogenesis. New neurons are created, but they seem to be in very limited regions, specifically the hippocampus, and they're very small. And for reasons, apparently about 99% of the new neurons that are created die off. So it's sort of a massive amount of competition. And we still don't understand its functional role in either normal or dysfunctional behavior. So let me summarize the sort of the brain component. Genes contribute to learning and memory, somewhere about sort of 50% at sort of a rough approximation of variance compared with environment. Brain development is responsive to environmental stimuli throughout. Both neurons and synapses seem to be overproduced and then pruned more so. So it seems to be more pruning than there is of sort of creating what you need. And then that echoes evolutionary theory. In other words, evolution doesn't sort of work by creating the type of beak that a bird needs for a particular island. It sort of overproduces birds, at a particular generation, and all the birds die off except for those that have the right beak to get at the particular insects on that island. Adolescence is considerable development of the frontal lobes at synaptic contacts, myelination, and changes. And gender differences, which are there in various degrees, tend to sharpen in adolescence and leading to differences in brain anatomy and some gender differences in behavior. And aging is associated with various sorts of brain deterioration at various rates, and Jesse will talk a bit more about that. So let me just talk finally about two different disorders that are linked to understand something about what we understand clinically between, and the reason we sort of chose this is that Down syndrome is primarily associated with early development problems, Alzheimer's with late stage, and this lecture, this chapter is about sort of the broad lifespan. And so we can see that there are some commonalities here. Once again, PowerPoint has failed me. So
into a debilitating condition. The connections are not correct and the processes aren't correct and there's some sort of deficit.
The directions for a human being are written in code, 3 billion letters long.
is that too loud?
These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Just as twins can be the result of unusual circumstances at conception or in early development, so can many kinds of birth defects. Early errors in the number or structure of chromosomes can lead to birth defects like Down syndrome.
About Down syndrome, the human body is made up of cells. Normally, the nucleus of each cell contains 23 pairs of chromosomes, for a total of 46. In babies born with Down syndrome, each cell contains an extra chromosome, chromosome number 21, for a total of 47 chromosomes in each cell. This condition is usually caused by an error in cell division in the earliest stages of development.
Almost 50 years ago, early genetic researchers discovered the presence of this additional chromosome in people with Down syndrome. They assumed that it alone caused the physical and developmental disabilities shared by people with Down. But today, our increasing ability to look at specific genes within each chromosome is giving scientists a clearer picture of what causes the condition. About 1 in 800 to 1,000 births results in a child with Down syndrome. It is the most common of all chromosomal abnormalities. Although individuals born with Down syndrome have physical and cognitive disabilities ranging from mild to severe, they are able to lead full lives with good medical care and education.
Good morning, Esther. How are you? Good. Good. How are you doing? Not too bad.
Individuals with Down syndrome can often have lifespans into their 50s and perhaps more. The aging process, though, is somewhat accelerated. Individuals with Down syndrome are at much higher risk for having an Alzheimer-like condition develop in their central nervous system, in their brains. And this leads to a decline in some of abilities that may begin as early as their 20s or 30s.
So what is it about this extra chromosome that affects the brain of people with Down syndrome? Scientists at the University of Wisconsin in Madison are using the latest tools in genetic research to look at each gene to better understand what happens in development and why.
We started a study looking at post-mortem fetal tissue collected from Down syndrome patients, where we knew that the fetal tissue had Down syndrome. And we isolated stem cells from that fetal tissue. We asked the question, are there any genetic differences in the human stem cells grown from Down syndrome when compared to non-Down syndrome stem cells?
The use of fetal stem cell research is controversial, but it's giving scientists an unprecedented window into early human development. Before human stem cells were available, the only way scientists could look at the genes that cause Down syndrome was by studying mice. But mice don't even have a chromosome 21, making the research a far less accurate gauge of what happens in humans.
I think one of the key developments of modern medicine and biology is the mapping of the human genome. And the other key development is the ability to grow stem cells in the dish. And those two things have come together. And I think by locking those two things in, we can learn an awful lot about biology and these diseases.
When scientists grow stem cells in the lab, they use the ability of the cell to replicate itself. Genes give each cell the directions it needs to determine what kind of cell to become. Through modern technology, laboratory scientists can turn genes on and off. This allows researchers to control the development of these cells.
Using stem cells that were programmed to make brain tissue, Dr. Clyde Svensson and his team grew these neural stem cells into balls of brain cells called neurospheres to help them find out how the cells replicate.
And this, Skip, we're sort of moving further and further away from the learning and memory, but that gives some background. So congenital formulation occurs equally often in boys and girls. One sees retarded speech, an extra copy of chromosome 21. And during embryo formation, a parent's mother usually fails to split properly. Brain size is generally average at birth, but there is growth in some areas. But growth in some areas is stunted, so particularly the hippocampus, the frontal cortex, and the cerebellum. Then have profound deficits, particularly in hippocampal-dependent memory abilities. So young adults with Down syndrome perform at about the level of a 5-year-old in terms of hippocampal function, and especially much worse at hippocampal. So here's looking at words recalled. I'm not quite sure what this is saying. Mental. What? I see. I'm sorry, I misunderstood. I missed that. So this is the Down syndrome adult at overall sort of age. An adult with Down syndrome is equivalent to about a 5-year-old on mental age, however that's sort of measured and approximated. If you look at Down syndrome adults or 5-year-olds, you see somehow similar amounts of words recalled, or actually worse words recalled. So the idea is that they're doing, they're overall at about the level of a 5-year-old, they're doing particularly worse on things that are hippocampal dependent. Okay, you can breed mice that creates an approximate version of Down syndrome, and they have spatial memory deficits. So again, suggesting some sort of some link between sort of hippocampal function and this genetics. If we look at Alzheimer's disease, it's a form of progressive decline. Affects about 4.5 million people, as many as 50% of people over age 85. Its earliest symptoms are, again, in sort of episodic memory, forgetting recent information, so learning episodic memory. Later on, declines in semantic memory and other functions. What this shows here is marked impairment. I've got to be careful. On trials, we have a 10-word list. 80 patients are doing overall much worse than those who are healthy, but that the deficit is exacerbated when there are delays involved. So learning impairments overall, particularly when there's sort of delays, again, we see this sort of these delay relationships being critical in hippocampal function. Its primary factors are, at a physical level, are amyloid plaques, deposits of beta amyloid, and neurofibrillary tangles. The amyloid plaques are throughout the cerebral cortex. The tangles are primarily in the hippocampal region. And let me show you one more. I'm not even going to assume that's going to work.
The human brain is a remarkable organ. Complex chemical and electrical processes take place within our brain that let us speak, move, see, remember, feel emotions, and make decisions. Inside a normal, healthy brain, billions of cells called neurons constantly communicate with one another. They receive messages from each other as electrical charges travel down the axon to the end of the neuron. The electrical charges release chemical messengers called neurotransmitters. The transmitters move across microscopic gaps or synapses between neurons. They bind to receptor sites on the dendrites of the next neuron. This cellular circuitry enables communication within the brain. Healthy neurotransmission is important for the brain to function well. Alzheimer's disease disrupts this intricate interplay. By compromising the ability of neurons to communicate with one another, the disease over time destroys memory and thinking skills. Scientific research has revealed some of the brain changes that take place in Alzheimer's disease. Abnormal structures called beta-amyloid plaques and neurofibrillary tangles are classic biological hallmarks of the disease. Plaques form when specific proteins in the neuron of the cell membrane are processed differently. Normally, an enzyme called alpha secretase snips amyloid precursor protein, or APP, releasing a fragment. A second enzyme, gamma secretase, also snips APP in another place. These released fragments are thought to benefit neurons. In Alzheimer's disease, the first cut is made most often by another enzyme, beta secretase. That, combined with the cut made by gamma secretase, results in the release of short fragments of APP called beta amyloid. When these fragments clump together, they become toxic and interfere with the function of neurons. As more fragments are added, these oligomers increase in size and become insoluble, eventually forming beta amyloid plaques. Neurofibrillary tangles are made when a protein called tau is modified. In normal brain cells, tau stabilizes structures critical to the cell's internal transport system. Nutrients and other cellular cargo are carried up and down the structures called microtubules to all parts of the neuron. In Alzheimer's disease, abnormal tau separates from the microtubules, causing them to fall apart. Strands of this tau combine to form tangles inside the neuron, disabling the transport system and destroying the cell. Neurons in certain brain regions disconnect from each other and eventually die, causing memory loss. As these processes continue, the brain shrinks and loses function. We now know a great deal about changes that take place in the brain with Alzheimer's disease, but there is still much to learn. What other changes are taking place in the aging brain and its cells? And what influence do other diseases, genetics, and lifestyle factors have on the risk of developing Alzheimer's disease as the brain and body age? Scientific research is helping to unravel the mystery of Alzheimer's and related brain disorders. As we learn more, researchers move ever closer to discovering ways to treat and ultimately prevent disease.
So that's actually, I thought, a beautifully visual representation of something I've been reading about and hearing about for a long time, the amyloid plaques and the neurofibrillary tangles. Verification of tangles, this statement now is sort of somewhat in flux. Originally, you could only see these tangles, plaques and tangles, in autopsy. Now they're beginning to develop imaging that hasn't been fully verified and validated, but more and more validation suggesting that we may be able to actually image these in the brain, such that 10 to 20% of people who are given probable AD based on all their observable behaviors may well have other disorders. There are several genes that are implicated in Alzheimer's, and what we know most about is the genetics of early onset. So there are people who can get Alzheimer's in the 35 to 50 range. It's incredibly early. In this case, it's very rare. It's about 1% of people with Alzheimer's have this early onset, but those who do have it usually due to several different particular genetic variants. And some of the stuff that we're doing now, Jesse is running, that we're doing now with UCLA is looking at some of these families. And we've identified several others in the West Bank. I picked these two disorders, Down's and Alzheimer's, because there are connections between them. So chromosome 21, which has been directly implicated in Down's, contains the APP, which has been implicated in Alzheimer's. By age about 35 or 40, most people with Down syndrome develop plaques and tangles. So they're getting essentially, they're in addition to whatever the developmental disorders, they also have early onset Alzheimer's. And half of Down patients show memory decline and other symptoms of AD. And this already note that Down's kids already have hippocampal deficits. So when you add on to that, the sort of early onset of Alzheimer's, it's obviously quite catastrophic, but others don't. So clearly Down syndrome isn't uniform. So in summary, Both Down syndrome is a condition of babies born with an extra copy of 21. They have cognitive impairments, particularly those that relate to hippocampus, frontal cortex, and cerebellum. In Alzheimer's disease, plaques and tangles accumulate in the brain. Memory symptoms are prominent. Again, consistent with the early deficits are in hippocampus and nearby medial temporal lobe. And there are several genes that may contribute to risk for the common late onset form, but they're all small risk factors.